Kakuna no sencha, Chana Poka Sencha, Kilo Pita Yomota, Don't Pota Itopa, Hilango Wapota, Watutango Watopa, Omarolo Matopa, Toandi Kamokoma, Chapotolon de Puma, Topo Kitoko Koma,
has white flowers and some others has blue flowers. It is the same species, so it shows variation. It either has white flowers or blue flowers. But when we have a crossbreeding between the white flower and the blue flower plants, all the offspring was blue. That helps us to see that this flower color is controlled by the dominant allele blue. My explanation is the F1 generation all shows only the dominant allele, which is blue. Use the symbols capital B, small b to represent the two alleles. So each parent was capital B, or small b, small b. And the genotype of the offspring will be one from there, one from there. Because during meiosis, gametes form and there's only one allele per gamete. And there we have the offspring. When we make a genetic diagram, when we cross one of the blue flowers with a white flower, the blue flowered offspring. So we use the heterozygous blue with the white. That is the phenotype. The genotype is capital B, small b, cross with small b, small b. The gametes will form during meiosis, and my gametes are in circles. Fertilization shows capital B, small b, small b, small b. The phenotype of the offspring is blue, blue, or white, white. So I can either have a heterozygous blue offspring or a white offspring. If they ask the ratio, this is the ratio. If they ask percentage, 1 out of 2 is 50%. Then we have the cup of a maize plant. And remember, when we draw a graph, this is X, this is Y. This is independent. We had a 7, a 8, a 9 cup. Then we count how many was there. That was 7 centimeters. 3 of the cups was 7 centimeters. 10 was 8 centimeters, right? And we draw a histogram. You will get a mark for having length of cup here and number of cup there. We did not sort this into ranges because it was a small amount that we were working with. We did not start at 1 centimeter and 100 centimeter. No, we had only 8 different. Shortest extreme was 7 and the largest was 19. Therefore, we could have written it like this. So you get for axis the name with its unit and the name with its unit on X, on Y. That is your axis mark. Then scale so that that distance is the same. And look at the 7. It's in the middle. Now, we write it in the middle now for the name of the column. Your column, you get your marks when they are attached, same width, each column, and draw with a ruler. Attach because it's a histogram, no spaces. Draw with a ruler, that will give you your mark for columns and the same width. And then for your plotting, that was accurate. That is where you get your marks from there. We say it is a continuous variation because there's a smooth gradation, no distinct categories. Or we can say there are extremes with intermediates. From 7 to 19, there are all the different lengths of cup sizes. The environment can play a role. The amount of light, minerals, water, temperature will or can influence the size of the cups. Explain how the type of variation shown by the maize cobs differ from that shown by the blue and the white flowers. They were discontinuous variation. You have to mention the name, describe it, or use the name to refer to it. And then we describe what do we mean with discontinuous. There's only blue or only white. No in-betweens, no intermediates. There's distinct categories. So I hope you got that answer. Grade 12. Now we can continue with our mutation lesson. Now that we have covered, there are variations. Variations is caused by the environment or by the genes. We have two categories, continuous. We start with one extreme, smooth gradation to the other extreme and all the in-betweens. Continuous, draw a histogram. Then we have discontinuous, distinct variation. Now, but in this variation, there can also form mutations. What is a mutation? A mutation is a sudden and unpredictable change in the genetic material of an organism. Now you remember we had three ways how a gene can change. A gene can change by if one of the bases is out, is deleted, or one can just be changed. It was a guanine, now it's a cytosine. So changed or deleted, altered or changed or deleted, or if a sudden an extra one appear. That is when the gene change. Now, and when the gene change, the order of the bases change in a gene, the recipe outcome of that protein will be wrong or different. The one mutation that we talked about was cystic fibrosis. You remember that gene that makes the mucus watery and not too sticky. If it's a faulty gene, it's a sticky mucus and prevent the uptake of oxygen. You got that one? Okay, that is gene mutations. And then we also have chromosome mutations, where there is something wrong with the chromosome in its completeness. So gene mutation, this sequence of the bases not copied precisely. And that changed the formation of the protein. Hemophilia, prevent blood clotting, that protein is wrong. Or albinism, where melanin, the part in the skin that causes the color of the skin, that brings color to the skin, but also protects the skin against the UV light, is not present. And then we have albinism, like we can see in this line. That is a gene mutation, which means either the gene is altered by having one or more of the gene bases changed. One is removed or an extra base, nitrogenous base is added. That's a gene mutation. We also add here cystic fibrosis. Now, is something that we can already see as mutations. We did that during our genetic problems. What about color blindness? It's also a defective gene on the autosome. The sex chromosome is also called the autosome. Now, that is also gene mutations. There we see a gene mutation where there's an overproduction of melanin. And here, very interesting, the two variants, overproduction of melanin and an underproduction of melanin. And it is not a new species, it is just a variation in the color. This is a lion, an African lion, but there's a variation in color, overproduction. And you know what happens if there is a lion like that? They all want to buy it because it's a very unique color variation. And if they start to breed with this male, there will be offspring that also look like this lion, like this male. So when we look at a chromosome mutation, it means there's a change in the structure of the chromosome or the number of chromosomes in the organism. Remember, the chromosome number is fixed to a species. How many in a human being? 46 chromosomes. If that number of 46 change, it is a chromosome mutation. It can also change the arrangement. And the example that we are going to study is Down syndrome. Now, Down syndrome is where we see when the baby is born, we see a flattened nose, slanting, upward slanting eyes, and interesting, the fingers are very short, especially the little finger, and there are this line right through the hand. Is also an interesting fact of Down syndrome. The toes that are widely apart, first and second toe, and lots of creases on the foot is a sign of Down syndrome. But what is different in the Down syndrome? It is pair number 21. If we look at the karyotype, you remember the karyotype is where all the chromosomes from the father and the mother are sorted from the largest to the smallest until we reach the 23rd pair. And now in Down syndrome, we see that pair 21 is not a pair, but we have 1, 2, 3. And therefore they say it is trisomy 21. They also call Down syndrome trisomy 21 because the mutation is visible in the arrangement of the pairs. It is not a pair in 21, but it is 3. What happens is during meiosis, we have the separation, and then they form two gametes. Actually, during meiosis, they form 4 haploid cells. But during this formation, the pair do not split. And where we have, the male has 44 plus the X and the Y chromosome in its genotype, and the female 44 plus XX. Now, they separate, and instead of having 22 plus X. There's 23 in the one chromosome, and in the other one, how many will there be? There will be 21 plus X. You see? But that one is not viable. So we ignore that one. That's not enough to have fertilization even taking place, or rarely take place. So if this is now a normal X chromosome, X sperm, and this is the other sperm with the Y. If we now have fertilization and this chromosome fertilizes the 23, now we have 24. Can you see? That is an extra chromosome. So this will be a female, and this one will be a male. If this sperm fertilizes with that extra chromosome. That happens when we have meiosis in the female. In this case, meiosis in the female, instead of having the 23 pairs splitting one to that side, one to that side during meiosis, it's not moving. 2 is moving to this side and none to that side. And then there are 21. So at chromosome 21. And there we see if there is an extra chromosome in the ovum. And that is more often the case, that there is more chromosomes in the ovum than in the sperm. The age of the mother can cause Down syndrome, the chance to have Down syndrome, to increase. If the extra chromosome is in the sperm cell, we will see that during the formation, we will have 23 plus X and 21, because these two must give me that number again, plus Y. You agree? And the female will be 22 plus X, 22 plus X. This one will give me now a female. If these two fertilize, I will have a female with Down syndrome. But I will not have a male with Down syndrome. There's no possibility to have a male with Down syndrome unless this 23 is now the one with the Y. You agree? If we have the Y there, I can have a male with Down syndrome. But if I have an X there, I will have a female with Down syndrome. I would like you, grade 12, to take your textbook and to check how they explain Down syndrome. Describe mutation as a source of variation as shown by Down syndrome. During the formation of the gametes, the sperm or the ova, 2 chromosomes of pair 21 fail to separate. The gamete has an extra chromosome. The egg or sperm can have now 24, 23 plus the autosome. Now, 23 plus 1 is 24. Okay, 24 instead of 23. After the zygote will form, 23 plus 22 will give us 47, 45, 46, 47. You see, this is equal to 47 because there we add the autosomes, the 6 chromosomes. Good. That is Down syndrome. And it is important that you know all the features of Down syndrome. Please do the attached question to see whether you do understand this or not. Read through this few pages, and then we have what can cause mutations to take place. Radiations and chemicals can cause mutations. Remember, cancer is a mutation. Why? Because the cell do not have the ability to stop dividing. It keeps on dividing, which means the message, the command for the cell to divide, to stop divide and start with its normal functioning is wrong. That will be a message from the genes that is wrong. So cancer is a mutation. When something can cause cancer, we say it's a carcinogen. You remember that word. But if we look at other things that can cause mutations, and it is a spontaneous reaction that takes place, we say mutagens are factors that cause mutations. UV rays, X-rays, gamma rays, alpha rays, beta rays. All those rays can cause the mutation. We know that the UV rays from the sun is the one that causes skin cancer. Then the tar in cigarettes can also be called a mutagen. Alcohol can be called a mutagen if it causes cancer in the stomach or wherever cancer is caused due to an overconsumption of alcohol. Now, we say the mutagen will not cause it, but it will increase the rate of the occurrence of a mutation. A mutagen will increase the rate of the occurrence. And with that, I want you now to please complete your worksheet.
We ready? Across Africa, in homes, towns and cities, men, women and children will stop. My heart is pumping just thinking about it, and ready themselves in front of their television sets, with hope in their hearts, and wait for the game to start. There is nothing bigger or better. On these days, we'll cry, scream, hurt, and dream. Hundreds of cities, millions of souls, united by football. It's the world's game.
Here we go! Glorious! Out of this world!
Stay connected to DSTV to watch the world's best football.
interesting work today. Let's continue with variation. We are still busy with genotype, what is determined by the genes, and phenotype. Eventually, what you can observe in a person was in his genes. Sometimes we know the genes can be masked off because there's a recessive gene, and then it is not visible to see in the characteristics. But eventually, we can see a lot of variation in a specific species. When we look at this picture that we have here, is that we can see these birds are all golden finches. They are of the same species, but we have a lot of color varieties between them. They are the same species, but there is a lot of differences between them. And then, of course, the human race. We know that there is so many variants in the humans. We must understand that with variation, we refer only to the differences between individuals of the same species. That is all that we are using when we use variance in when we use variation in a species. That is what we are referring to now. Now, as I've said, this variation is caused by what do we see in an individual, what do we observe? And sometimes it is also not visible what is a person's blood type, but it can be A, B, O, or AB. But what causes that blood type? The blood type is caused by the genes. Now, the blood type is caused by the genes. When we look at the phenotype, it can be caused by the genes, but also by the environment. Because I can have a nice dark skin, but it is because I am all day in the sun. That is environment. My genes tells me what is my blood group, and the environment cannot change my blood group. That is only by the genotype. We have two different variations. It can be continuous variation, which means there is no distinct category. It is smooth gradation. It can be caused by the genes and the environment, or by both. What am I talking about? I'm talking about height, skin color, intelligence, and there are many more variations. Let's look at first height. Here I have head of states, some not in the current running, but we can see that they start at 5 foot 2 inches and they increase. It is a smooth gradation. There's not a distinct category. If you are head of a state, you are this length. No, it does not work like that. Another example, if we take the smallest feet in the school and we put then the next size, we will have a lot of feet next to each other. And they will just increase a tiny bit. Smooth gradation. No distinct category 4. Grade 5 feet are 12 centimeter. That's a distinct category. No distinct category for continuous variation. We also can have the same with eye color. We have a lot of different variations in eye color. And